And these first 3 screens, you can pause this video and you can write down the notes at the top of those pages. So enzymes are important molecules in biology. They're proteins that speed up chemical reactions in the body. Without them, many processes of life would not happen. They're very specific in their functions. Each enzyme only has one reaction that it can help. So you have thousands of different enzymes for all the thousands of different reactions that happen in your body. Enzymes are not changed when they perform their function. This means that the same enzyme molecule can be used over and over and over again. So that's really helpful because that means you don't have to make new ones every single time. You can just use them over and over again. Some enzymes may even be used repeatedly on the same set of small molecules to build a long chain of repeating subunits. So if your body is building a really big protein, it's going to use the same enzyme and it can string together a bunch of amino acids to make that one protein. As with all proteins, the shape is what determines its function. An organism has the ability to make many different enzymes, and each enzyme has one particular function. And the shape of it is super important. Sometimes high temperatures or extreme pH values may affect the shape of an enzyme. This is called denaturing. That means to take away the natural shape of something. It can make an enzyme work less effectively, or it can destroy it and make it useless altogether. Notice that this says high temperature. It doesn't say low temperature. So low temperature will just slow chemical reactions down, but it doesn't destroy enzymes. You can't freeze an enzyme and denature it that way. So you can slow reactions down with cold, but only high temperatures would actually destroy the protein of that enzyme and denature it and render it useless. So we're going to look through some animations that help us learn this. First, why enzymes? And this goes with all of the pages in your packet. So first, we're going to go with without enzyme. So here we have some simple sugar molecules. And you might remember that a simple sugar molecule is called a monosaccharide. And we want to put these two simple sugar molecules together to build a disaccharide. So without an enzyme, they bounce off of each other and they haven't quite lined up to bond together yet. So they can bounce off each other a long time until they reach at exactly the right angle to actually connect together. And since they didn't do that, it's just going to take like a really long time. So with an enzyme, this is the enzyme, and see how it has this active site that these guys can fit into. That allows a place for this reaction to happen. So they come in, they fit together, the reaction happens to connect these monosaccharides and form a disaccharide, and the reaction happens much more quickly. So this forms a bond, disaccharide, they're linked together, and now everyone is happy. You'll notice that when the double sugar molecule leaves, again, this is called a disaccharide, the enzyme is unchanged. It still has the same shape it had before it helped with this chemical reaction. So now this enzyme can help more single sugar molecules become double sugar molecules again and again and again. Specificness. So here we have two enzymes on the screen. One is green, one is yellow. You're going to want to make sure you're using your colored pencils or highlighters here to show the difference. But you can see that the active site is actually different too. So now say we have this double sugar. Instead of building it up, we want to break it down. So as you can probably guess, this is not the right fit. This is. So it won't fit. So if it won't fit, this enzyme clearly won't be able to help do anything to this double sugar. This enzyme must be made for some other reaction somewhere in the body. But this double sugar can fit here. It can fit, and it will help the sugar molecule be digested, and now we're going reverse, so now it's breaking apart into monosaccharides. So that'd be an example of something that happens probably in your small intestines, breaking down some carbohydrates that you ate. Now, here's two different sugars that you want to build together, it looks like, because they're separated. And you can see this one does have the right shape, so the green enzyme can fit those together, make that bond, and now we have a different disaccharide. Obviously, the yellow one would not be able to help this one. So every single enzyme has a different substrate that it can help, and that depends on the shape of its active site. Reusing enzymes. So enzymes can be used again and again and again for separate reactions or for one continuous reaction, which is this example. So you can see that these monosaccharides came in and formed a bond. And if I press play, this one kind of scoots over and lets this one come in. And now we have a trisaccharide. Again, that means 1, 2, 3 sugars. Here comes in another monosaccharide. Now we have a 4 sugar carbohydrate, and that just keeps going. And we're making this really long polymer. Since this is a sugar, this would be called a polysaccharide. So we can build really long proteins like this, chains of amino acids called polypeptides. Starch is a type of carbohydrate, so that's polysaccharides. This is even how we put DNA together, deoxyribonucleic acid. And that's going to be made of nucleotides. Last but not least, when bad things happen, denaturing. So shape, obviously super important. These guys can fit in, we can make a disaccharide, hooray. But now we add heat to this protein enzyme. And when we add heat, the shape changes. And now that the shape has changed, the active site has changed so much that these monosaccharides really can't fit in there anymore. And since they can't fit, you can see they're not close enough together to make the chemical reaction work anymore. So this enzyme is pretty much useless. So they won't react unless they get lucky and bounce together at the right spot, which is possible. That just takes a lot more activation energy, a lot more time. So unless there's some other enzymes that aren't denatured, this reaction will happen really slowly. One other thing besides heat that does denaturing is when the enzyme is out of its correct pH range. So if it's supposed to be in an acidic pH and instead it's thrown in a basic pH, that could denature the enzyme as well. So I hope that was helpful. Please watch this video as many times as you need to, pausing it at different places to finish your organizer on page 54 to 60. And if you ever get access to a computer or Chromebook that has Flash, you can click through this yourself. So I hope that was helpful. And let's see how I stop this video. Let's see.